In the previous video, we talked about the first step in the embryonic stages, which is fertilization. In this video, we will talk about the next step, which is cleavage, and that's the kind you're thinking of. Within this step, we will 1. Talk about what occurs during cleavage, 2. Figure out what cell cycle phases occur during cleavage, and lastly, we'll see how cleavage differs among different animals, such as mammals, amphibians, and birds. Cleavage is a period of rapid cell division, and occurs without any growth in size of the cell. During cleavage, the zygote cytoplasm is divided into smaller cells known as blastomeres. This group of blastomeres produces a blastula, which is a hollow sphere of cells. The hollow center, which is a fluid-filled cavity, is known as the blastocele. The process of cleavage can be seen in these 4 steps. So in step 1, you see the fertilized egg, also known as the zygote. Then we move to step 2. This is known as the 4-cell stage. So you begin to see the zygote divide into further cells, which are known as the blastomeres. In step 3, you begin to see an early blastula. So you see the cells have divided further, you get more blastomeres, and you begin to see that fluid-filled cavity, the blastocele, seen in the bottom left-hand corner of within the blastomeres. And lastly, you have the blastula. So you have the blastomeres in that full ring, and you have the fluid-filled cavity in the center, also, again, known as the blastocele. Next, it is important to note that during cleavage, only the DNA synthesis, or S phase, and mitosis, or M phase, occur. Because G1 and G2 are skipped, the size of the zygote does not change. During the S phase, DNA, or chromosomes, are duplicated and makes 2 identical copies of itself. During the M phase, mitosis and cytokinesis occurs. Mitosis is the division of chromosomes, or DNA, into 2 separate nuclei, and cytokinesis is the division of cytoplasm into 2 new cells. During the G1 phase, the cell gets bigger and prepares for completion of S phase, and during the G2 phase, the cell grows more, copying more organelles such as centrioles, spindle fibers, and certain proteins, and reorganizes within the cell, which allows the cell to be ready for the M phase. Because of the exclusion of these 2 phases, as you can see here, the cell will not grow bigger during cleavage. Cleavage among animals differ. For species with yolk involved in their egg, it has been found that animals with yolk have a predetermined, specific pattern division of cleavage. And due to yolk, the cleavage within these eggs are asymmetric. The yolk is mostly found in the vegetal pole in comparison to the animal pole. So as you can see here, you have the vegetal pole on the bottom and the animal pole on the top. So what this means is that the yolk will mostly be found here, and in comparison to the animal pole, there might be just a slight amount of yolk within there. Also, the animal-vegetal axis has a predetermined anterior-posterior axis in place of the egg. So in other words, the animal hemisphere makes up the anterior axis, and the vegetal hemisphere makes up the posterior axis, as seen here. So again, the vegetal hemisphere will be predetermined with the posterior axis, and the animal hemisphere will be predetermined with the anterior axis. So before we move on to the stages of cleavage, we have to talk about the gray crescent, what it is, and why it's important. So once the sperm and egg fuse together in fertilization, as seen here. So again, here's the egg, here's the sperm, and this red dot is the sperm entry. The animal hemisphere rotates towards the point of sperm entry, as shown. So again, once they fuse together, the egg rotates forward. This rotation is known as the cortical rotation. And because of this rotation, an area known as the gray crescent is shown. And the gray crescent is the future dorsal side of the organism. So the dorsal side is the back side of an organism. So think of a dolphin, for example. It has a dorsal fin, which is on the back side of the organism. And now we have the actual stages of cleavage for the egg. First is the first division of the egg, which is cleaved vertically and forms 2 cells. The blue line is the vertical cleavage, and here's 1 and 2 cells. The next division is a second vertical cleavage, and this forms 4 new cells. So now we have 1, 2, 3, 4 cells. And this black area right here is the gray crescent that we talked about before. After this, the third division occurs, this time splitting the zygote horizontally, as shown. So here you have the blue line again, split horizontally. So now you have 8 cells. So 1, 2, 3, 4, 5, 6, 7, 8. And that's why it's called the 8th stage. And finally, the blastula is formed, ending the cleavage step. It is important to note here that the uneven distribution of cleavage here is due to the uneven distribution of yolk among the cells. And the last thing I want to mention is the two different types of cleavage. So there's meroblastic cleavage, which is when there is more yolk in different eggs, such as birds, reptiles, and fish. Because of this, it is more difficult for cleavage furrows to pass through the entire egg, and only the animal hemisphere, which lacks yolk, undergoes cleavage. Then there is holoblastic cleavage, seen in amphibians and mammals, which occurs in species with little yolk. Here, the cleavage furrow will pass through the cells fully. So before I go, I just want to say thanks for watching. Please like, subscribe, and leave any questions in the comments in regards to this video or any videos you guys would like to see in the future. Thank you.